Okay, so I'll just do a couple of housekeeping things here.
Okay.
So welcome to everyone. Thank you so much for joining us on our weekly research fight that is in place of the annual INKY conference that this year was supposed to happen in Toronto, but unfortunately due to COVID, we are doing everything online. But it means that we've managed to have some longer conversations and some really great presentations so far. Today should be in that same category as we have Nadine Vogt here to present on her current PhD research on raccoons and the epidemiology of various zoonotic diseases and foodborne pathogens, as well as associated antimicrobial resistance. And so Nadine is a graduate from the Ontario Veterinary College and currently doing her PhD in the Department of Population Medicine. And hopefully we'll all have a little bit better understanding about why you shouldn't be taking baby raccoons home after this presentation. Just as a reminder, please do put yourself on mute. I will be muting everyone from my end as well, but just to make sure we don't have any of that background noise that makes it impossible for me to eliminate when I am then trying to splice everything together so that we can post it for people who weren't able to be here. We will have a question and answer session at the end with Nadine, so if you want to submit your questions to either Andrea or I via the chat rather than the question section, that means that we only have to go looking in one place for them. So Nadine, it's all over to you.
Great, thanks so much for the introduction. And thanks to Jasmine and Andrea and other conference organizers for this opportunity. Just bring up my main slide here. So the main focus of this presentation will be on some recent work on Campylobacter that I was involved in as part of my PhD thesis. I would like to acknowledge my co-authors here on this Campylobacter work. They're listed here, as well as the many other researchers, students, and staff who've been a part of the overall greater project. Finally, thanks to Elizabeth Bett for the use of her lovely photos. So I'd like to begin with an outline or roadmap of the talk, because we'll be covering a number of different but related topics. So first off, I'll begin with some of the reasons why raccoons are particularly interesting and important wildlife species to study in the context of zoonoses. And then next we'll move on to some of the details of the overall study design, including some of the field work and data collection methods. And then this section here, what was I going to say? We'll discuss some completed work that's already been published. And so the reason for doing this is to help provide some context for the Campylobacter study that will be the bulk of the presentation. So hopefully we'll help to provide an overview of the different zoonoses in raccoons. So we'll be looking at previous work in this population of raccoons, looking at Salmonella, looking at generic E. coli, associated antimicrobial resistance in those bugs, and then Clostridium difficile, or C. diff as it's better known colloquially. And then at that point, we'll come to the bulk of the presentation. So if my mouse doesn't stick, the Campylobacter study, and that was work I was directly involved with. And then that'll put us in a position to look at all of these studies in context together and hopefully draw some general conclusions about zoonoses in raccoons. And then finally, I'll finish up with a discussion of some of the ongoing and upcoming work, which is part of my PhD thesis that utilizes whole genome sequencing data. Okay, so probably most of you understand the One Health approach and why it's important to study wildlife. However, raccoons in particular are an interesting species and important species to consider in this field of research. Most of you know they're opportunistic feeders, so you'll find them rummaging through your garbage. They'll eat just about anything, whatever is available. They're, pretty intelligent species. They get into all kinds of places, and so when there isn't as much food available, they do have to roam a bit further in order to feed themselves. So they can have quite large home ranges. In the prairies, ranges of up to 50 kilometers squared have been reported. That's not so much the case for Ontario. Ranges are more in the area of 4 kilometers squared. But what that means is that they can travel from different types of locations. to others. So, for example, urban raccoons can end up in farmland areas that are in close proximity potentially. And it goes without saying, increasing interactions between humans and wildlife species are potentially a problem. With these shared environments, there's a potential for transmission of various pathogens between people and animals, between raccoons and pets, and all sorts of different sources in that manner. And so arguably, raccoons are kind of an underappreciated species in this field of work. There's been a lot of research looking at wild birds. Birds are highly mobile. We see them in all kinds of places. We see them on farms interacting with cattle. And so there's more potential routes of transmission, but you know, there's increasing work on raccoons in recent years. And so this study was a unique opportunity to look at raccoons in Southern Ontario over a number of years. So it's a very intensive project that took place from 2011 to 2013. The design of the study was a repeat cross-sectional study. And as you can see here, raccoons, this little guy, they were live trapped, and that was done from May through November. And the types of locations that were sampled, based on previous work in wildlife, that found that swine farms and farm environments were associated with higher prevalences of antimicrobial resistance or certain pathogens. We decided to look at, I shouldn't say we because I wasn't involved at that point, but it was decided to examine swine farm environments versus conservation areas to see if there, you know, maybe some differences there between the wildlife species on those types of locations. So these areas were sampled near Guelph and Cambridge in Southern Ontario, and animals were sampled up to once a month. So if they were caught more frequently than that, they were released, so as not to have negative consequences from too much anesthetic and that sort of thing. So animals were tagged and microchipped so that they could be identified, and the age and sex of the animals was recorded. And age was determined based on the size of the animal. So you can see here, this little guy, he's a juvenile. Another way that age was determined was looking at the dentition and wear of the teeth of the animal. Okay, I'm not sure. Oh, there we go. Okay. So, you know, further details about study design. So as I said, animals were anesthetized. For kind of a subset of the animals, paws were swabbed for Salmonella to look at the potential for mechanical transmission. But primarily the focus was on fecal swabs and, you know, biological kind of transmission via feces. So the different organisms that were sampled were Salmonella, Clostridium difficile, generic E. coli, and then for some of those we looked at the antimicrobial resistance, or AMR, and then Campylobacter as well. So to kind of flesh out the picture of what was going on, environmental samples were also obtained. So soil samples were taken from very close to the traps where the animals were trapped on both location types. And as you can see here, manure pits were sampled from swine farm environments, and then dumpsters were sampled in conservation areas. So in terms of the previously published work on this, it's actually part of Kristen Bondo's PhD thesis. We'll begin with Salmonella. We found an overall moderate prevalence of Salmonella. So, you know, about 1,000 animals were trapped in total, or sorry, 1,000 samples. So some of those animals were captured multiple times. So of, you know, 1,093 samples, 26% of them were positive for Salmonella. So that's considerable. And a similar prevalence was seen in those environmental samples. So that included swine manure pits, dumpsters, but the soil samples seem to have a slightly lower prevalence. And in terms of the serovars that were detected, many of them were overlapping with those serovars causing illness in people. Some of the most prevalent ones found in this raccoon population were Salmonella Newport, Salmonella Oranienburg, and Salmonella typhimurium. So in terms of looking at the risk factors, statistical modeling was done looking at, you know, whether factors like the location type had an impact. As I mentioned, the hypothesis was that swine farms would potentially impact the prevalence of Salmonella in raccoons. However, this was not seen. So there was no association between the location type and the occurrence of Salmonella in raccoons. However, there was an association with season, so that corresponds with the biological season. So whether raccoons are rearing their young and they're not moving around as much. especially for females, versus when the young are dispersing and they're out in the environment. And we also saw associations with year, the amount of rainfall, and raccoon sex, with males being higher. So although we didn't see an association with the different location types, when we looked in more detail at the serovars and antimicrobial resistance patterns, there did appear to be differences between the different location types based on those. Now, in the smaller mechanical transmission study, so this is a smaller subset of raccoons, about 400 samples. So a paw sample and a fecal sample were compared in each case, so they're paired from each animal. And in some cases, Salmonella was recovered from both sample types. In some cases, just the paws, and other cases, just the feces. But what this is telling us is that, raccoons are potentially transmitting Salmonella just via, their paws and walking around. Sorry, I guess I'm just having a little bit of trouble clicking here. Computer glitch.
Can you use your arrow key?
Yes. Oh, I can go to the bottom? Okay, great. Okay, so next, if we look at the findings from this population looking at Clostridium difficile, again, not all animals were sampled for this. This is a subset of the animals. Of about 400 samples, 9% of the fecal samples were positive. A number of different ribotypes were identified. Some of these are responsible for human illness. And based on animals that were captured on multiple occasions, it doesn't look like they're carrying this organism for very long at all. And similar to the findings with Salmonella, C. diff was not associated with the location type, so farms were no higher than conservation areas in terms of the prevalence. But when the researchers took a closer look at the different ribotypes and their distribution, that did in fact appear to vary. For example, ribotype 78 was significantly associated with farms. And finally, if we look at the E. coli work and the antimicrobial resistance in those, so the antimicrobial resistance testing was done with the CPARS or NARMS panel. I think it's about 15 different antimicrobials that are examined. So when characterizing the prevalence of resistance to at least one, this was very high in the manure pit samples, but 60% of samples were positive for some sort of resistance. The prevalence was a lot lower, and it was about 10% in other samples, so soil, dumpsters, and raccoons. And again, with the risk factor analysis, location type did not appear to predict whether AMR was higher in a certain location or not. But similar with what we were seeing with the other organisms, when looking at the extended spectrum beta-lactamases and the different resistance genes, those did appear to be much higher in conservation areas. And in fact, some of them were nearly absent in the swine farm locations, the location samples. So when looking at things in more detail, it does appear there are qualitative differences. So we're all familiar with this kind of image and this kind of situation. We see raccoons dumpster diving and digging through garbage and opening green bins all the time. And so, we're thinking maybe at this point that the dumpsters were a source of antimicrobial resistance determinants. But we need more information to look at this in more detail and to verify these findings. So now we move into the bulk of the study. So this is some work I was recently involved with, looking at Campylobacter. But just quickly before I delve into that, there was some recently published work using the data from this population by my colleagues Mitchell et al. And what they found was by looking at the genotypes of Campylobacter using comparative genomic fingerprinting. So that's a method that Ed Tabota and his lab developed out in Lethbridge, Alberta. So they basically assessed for 40 different presence or absence of 40 different genes. And so what they found was, they saw a high prevalence, 46% of samples were positive. It was carried transiently, so, less than 30 days at a time generally. And so the folks out there, they have a massive database. They've been collecting Campylobacter samples from different sources, including human, wildlife, livestock, all kinds of samples, and they've typed them using the same approach. And so when comparing the raccoon Campylobacter to these other sources, what they found was that the genotypes were really similar to livestock and human clinical infections in many cases. So 66% of the time, or 66% of those samples overlapped with those different sources. In addition, some genotypes that were specific to raccoons were identified, so these were not identified in any other source. So that was also interesting to note. Okay, so that brings me to my work. So the objective of this work was to build on the genotyping study and to figure out whether certain risk factors were more associated with the carriage of Campylobacter in raccoons in this population or not. So our goal was to assess, seasonal, climatic, location, annual, and then raccoon demographic factors, and whether these were associated with the isolation of Campylobacter from raccoons. So to do this, we used multi-level, multivariable logistic regression. So we're looking at the odds of isolating Campylobacter jejuni or not. And so we used multi-level just to address potential clustering. We had multiple samples from some of the raccoons, and we also had multiple samples from certain locations. So we included those as random intercepts. And I'll just note quickly here, the most predominant species of Campylobacter that we found in this population was Campylobacter jejuni. So for the purposes of statistical modeling, we just restricted our approach to Campy jejuni. However, we did identify some Campylobacter coli, but, a handful of samples. And then there were some samples that were untypable. But again, they were found in small numbers. So in terms of the independent variables we looked at raccoon demographic factors potentially. We looked at location type again, so looking at swine farms versus conservation areas. We also included the year and the season. Again, this is the biological season, so whether raccoons are breeding or their young are dispersing. And then finally, we looked at the different climatic factors, so the sum of rainfall and the mean air temperature, and these were taken over the previous 14 days prior to sampling. Okay, so I'll save you the trouble of looking at many complicated tables because in the final model, we ended up having 3 interaction terms and their main effects. And, for these variables, some of them had 2 or 3 categories, so it got very complicated very quickly. But essentially what we found was that rainfall, temperature, and season were all significantly associated with the carriage of Campylobacter jejuni. But the effects were really not consistent. So depending on what year you were looking at, rainfall had a different effect. So it wasn't consistently that, high rainfall always predicted a higher carriage or higher odds of Campylobacter carriage. So there were no overarching patterns that we could identify, even though these associations were identified. We didn't find any associations with those demographic factors. So age and sex did not predict the carriage of C. jejuni. So looking here at what exactly was included, we saw an interaction with the season and temperature, year in rainfall and year in temperature. And so that's telling us probably that, the environment is a very important predictor, of whether raccoons are exposed to Campylobacter or not. But that really varies depending on what year you're looking at and what location you're looking at. We did identify there was clustering by animal, so samples obtained from the same animal were more similar, so we retained that in the model. Okay, so together with the recently published work by Mitchell et al. With the genotyping data, what we're seeing is that raccoons are indeed acquiring genotypes of Campylobacter that are associated with human infections and livestock. But, based on our recent work looking at risk factors, it's unclear what the source is. It's possible, similar to the other work we've done or that's been done, that, we're missing some of those predictors. Maybe we need to sample different sources on the farms. For example, water is a well-known source of Campylobacter. So looking at proximity to rivers or nearby water sources or different sources on the farm itself. So to answer some of these questions, we'd like to delve into this a little bit deeper. And so my colleagues are going to use whole genome sequencing of these isolates to kind of, place things into context and compare these Campylobacter samples with the isolates in their database to answer some of these questions about transmission. So just briefly, I'd like to touch on some of the limitations of this work. So in particular for the Campylobacter study, we didn't account for possible confounding due to strain-related differences. So It's well understood that some subtypes of C. jejuni are more resistant to certain kinds of environmental exposures. Some are more resilient in the water. So, unfortunately, for the purposes of this exploratory work, we didn't account for that, but it may be an important thing to consider. As I mentioned, we may be missing predictors. So our model, we're exploring the different factors, but, perhaps our characterization of some of these things, such as the climatic factors over 14 days, maybe we need to look at a different time period and that sort of thing. And then finally, one limitation that is common to all of the work that I've just gone through is the selection bias. So this raccoon here isn't particularly happy to be in the trap, but there were definitely some trap-happy raccoons, as we call them, because they came back 6 or 7 times. There was one raccoon that came back 8 times, so clearly did not have an aversion to being trapped, was probably happy to get some free sardines. And so this definitely impacts the results because it's possible we're getting a subpopulation of raccoons that aren't representative of the greater population that's out there in Southern Ontario. So when we're looking at all this work together, trying to find some common threads, what we're seeing is that raccoons, although they didn't appear to be ill, they're carrying, a wide diversity of pathogens. These are known to cause illness in people, so that is a concern. Almost across the board, we saw that climatic and seasonal factors were important predictors of the carriage of these pathogens. And demographics sometimes were, sometimes weren't, depending on which bug we were looking at. So something I focused on with each organism was whether location type was predictive. And so what we're seeing is with the characterization that we have, the broad characterization of swine farms overall versus conservation areas, we did not see any significant associations, but that doesn't mean that, there aren't differences. On a qualitative level, differences were noted as soon as the data was looked at in a bit more detail with ribotypes, with serotypes, et cetera. So that more detailed information was suggestive of differences. And so in the future, we definitely need to use highly discriminatory methods and combine that with epi approaches in order to better address these questions. So I'd like to finish by just touching on some ongoing work that's part of my thesis. I've been fortunate to be involved in this project, coming on later in the project, and basically we're going to be we've already whole genome sequenced the Salmonella and E. coli isolates. And so we're going to look in more detail using core genome multi-locus sequence typing to try and figure out whether there's clustering between among the wildlife sources, whether there's overlap with other sources like livestock and human samples. We're fortunate to be able to compare this data with human clinical data that was collected in the same geographic region over the same time period. And so we can kind of get at some of those questions about, is there overlap? Are raccoons carrying Salmonella types that are also found in human clinical infections? And so hopefully we'll have something that looks like this eventually, looking at the different sources and how they kind of parse out, whether they're clustering together, whether there's overlap, and combining that with an assessment of the distribution of plasmids and resistance genes to better understand how antimicrobial resistance is moving within these different sources, whether it's by plasmid or clonal expansion. So there's a lot of interesting questions we'll be able to address with this kind of data. So very exciting work coming up. Finally, I'd like to acknowledge everyone that's been a part of this project. This is a huge undertaking. Thanks to NSERC for providing funding for the project. Thanks to my advisory committee, David Pearl and Claire Jardine. Field and lab support, this wouldn't be possible without them. This is 3 years of being out in the field, early mornings, dealing with unhappy animals in some cases. So thanks to everyone there. And then finally, thanks to everyone for these particular organizations for providing funding for scholarships. Oh, and then I guess I'll leave that there. And thanks so much for joining, and I'd be happy to answer any questions.
Great, thanks, Nadine. That was very interesting. So raccoons are readily acquiring the human and livestock Campylobacter, but has there been any real look at whether the transmission is going the other direction as well, so that the raccoons are actually introducing Campylobacter to humans and livestock?
I think that's a great question. I think this is like if I'm understanding correctly, it's a bit of a chicken an egg situation, I think oftentimes, and I'm always cautious of this, I don't want to paint wildlife with a bad brush and say, it's their fault. They're transmitting stuff to us, but, perhaps we're transmitting stuff to them and they're moving it around. So we're not sure which direction things are going in. And I'm not sure, that's a really great question to think about. Like, how would you address this? Who's starting this in a way?
Right. As with many diseases these days.
Yeah.
And then were other types of livestock production taken into account? So poultry or dairy or beef, and the proximity of these production types?
Are you thinking of specifically one of the studies, like Campy, or any of the?
Well, feel free if you have the information on all of them to answer. But
yeah, we hope to address that with my current work. But for the work I've presented today, up till and including Campylobacter, that kind of information wasn't taken into account. We were really just looking at whether the animals were captured on a swine farm. So we knew they were all swine farms, but it's possible. I think one of the locations actually had poultry on it too, but that was not accounted for in the modeling approach. And I think mostly just because this is just first step, it's most exploratory at this point. But in the work I was describing with Salmonella, we have samples that were obtained through surveillance. So they were livestock samples. So we have different types of livestock in Ontario, I think Wellington County and Waterloo region. So we will be looking at whether those are from poultry versus beef versus dairy versus swine. So hopefully we can address that question using those approaches.
Okay, great. And then you mentioned a One Health approach and the human testing for the pathogens within the same geographic location at the same time. And was this always planned, or was there an agreed to collaboration that had been foreseen?
I'm not sure. I know that when I came into the project, that was the plan, but I'm not sure that originally that was the plan. It just so happens that we're able to get this data, which is, an incredible opportunity to compare something in the same area in the same time period. So my feeling is that we've acquired that since, through collaboration.
Great. And anytime you can actually pull in as much data as possible, obviously as epidemiologist, I'm like, the more you know, the better. Though it can sometimes get overwhelming with a data overload.
Too many questions, right? Yeah.
And then you go off on all kinds of tangents. But so how do the results in the raccoons compare to feral or farm cats then?
To other, actually, I don't think we have those kinds of samples, but that would be really interesting. But you said cats, right?
Yeah, I did. So then that you could not only add on the livestock, but you can also add on companion animals. Let's go whole hog here.
That would be awesome. Yeah. Future directions, right?
Yeah, exactly. And so then with respect to the antimicrobial resistance patterns affiliated with the isolates, were the raccoon isolates related to the farm isolates?
So to be totally honest, I'm not super familiar with the details of some of this work. This is something I can get back to people about, I think. Is that right?
Sure. Yeah. So if you are able to go find that information and you send it to me, I'll include that then when I send out the next update for our next meeting. And does anybody have any further questions? We're now at the end of our time, but if someone had a very, very quick question, we could potentially ask it. So has anyone looked at similar information near the human hospitals at all that you know of?
Like wildlife near the hospital? Not that I'm aware of. I haven't systematically search the literature for that, but that would be really interesting because I know that there's a lot of affluent that comes out of the hospitals if they're near rivers that maybe is a potential source of exposure. Yeah, that would be really neat. I'm not sure.
Okay. Well, if anyone has any further questions for Nadine, please do feel free to email me and I will send them to her and get those answers out to everybody. So next week we have Sarah Robinson, who's going to be talking again about zoonotic pathogens, this time carried by urban rats. And having just moved away from Ottawa and being very happy to be in Alberta, where technically we don't have any rats, this is going to be another presentation, I'm sure, that makes my skin crawl a little bit. Anyway, everybody, thank you so much for joining us this week, and have a great week. Please do stay safe, and don't forget to wash your hands. Bye, everyone.